Today, we're diving into a really fascinating, almost radical new way of thinking about aging. What if you could actually slow down the aging process at a cellular level, not just cosmetically? Let's get right into the data.
So here's the central question, the paradox we're trying to solve. We've known for decades that caloric restriction has these incredible anti-aging effects in lab settings. The problem? It's incredibly difficult for humans to maintain in the real world. So, is there a way to get those same benefits, to essentially trick yourselves into thinking you're restricting calories without the restriction part? Well, the solution lies in a concept that scientists call a caloric restriction mimetic. It's not about eating less at all. It's about introducing a specific substance that signals to your body's deep cellular machinery that you're consuming fewer calories, which, in turn, flips on those same powerful pro-longevity pathways. Now, you might be thinking this agent is some exotic lab-synthesized molecule, but it's not. It's something far more common, something we've often overlooked for its true hidden function. We are talking about indigestible fiber, but not for its role in digestion, no, for its role as a potent biological signal. A landmark study published in Nature really laid out this specific protocol for this. And let's be clear, this wasn't just about adding a bit more fiber to the diet. In what they call the CF30Me diet, the researchers took a standard mouse diet and replaced a full 30% of the macronutrients with pure indigestible cellulose. And what's absolutely crucial here is that the vitamin and mineral levels were kept exactly the same. This was a very precise, very controlled intervention. So, what happened? The results were pretty striking. The group on classic caloric restriction ate 20% less and, as you'd expect, lost about 17% of their body weight. But the Fibermaxxing group, they actually ate 47% more food by volume, yet their body weight barely changed. And here's the most important part. When they analyzed their gene expression, their transcriptional profiles started to perfectly mimic the classic anti-aging signatures you see in caloric restriction. They were getting the molecular benefits without the deficit. So how is this even possible? How can eating more trigger a response that looks like eating less? Well, the mechanism doesn't actually start in our own cells. It begins with the trillions of microbes living in our gut. That indigestible cellulose, it isn't food for us. It's the raw material for what you can think of as your own internal microbial pharmacy. You see, our gut bacteria take this indigestible fiber and they ferment it. And this isn't just a waste process, it's an active manufacturing process. As the bacteria break down the cellulose, they produce a whole range of incredibly powerful signaling molecules, which we call postbiotics. And these molecules, they are the key to this entire process. Now, these postbiotic signals don't just stay in the gut. Oh no, they get absorbed into our bloodstream, travel all throughout the body, and they interface directly with our cells. They act like instructions, and they're capable of directly altering our epigenetic signature. That is, the very system that controls how our DNA gets expressed over our lifetime. This, of course, leads to the next critical question. If this dietary change is actually altering our epigenetics, can we measure it? Is there a way to objectively quantify whether we are, in fact, slowing down the pace of biological aging? And the answer is yes. The technology to do this exists, and it comes in the form of epigenetic clocks. These are highly sophisticated algorithms that analyze the patterns of DNA methylation. These are tiny chemical tags that attach to your DNA and tell your genes whether to switch on or off. By reading these patterns, we can calculate a person's biological age, which, as you can imagine, can be very different from their age in years. Now, among these tools, the DunedinPACE clock is widely considered the gold standard for this type of research. And it's unique. It doesn't just give you a static biological age number. Instead, it works like an aging speedometer. It tells you the rate at which you are biologically aging right now. A score of 1.0 means you're aging 1 biological year for every calendar year. Anything below 1, you're aging slower. Above 1, you're aging faster. It's an incredibly powerful metric. Okay, so that's the mechanism and the measurement tool. But how does this play out in humans? Let's move from the mouse model and look at the evidence from large-scale human studies. This is where we really connect the dots between diet and that epigenetic speedometer. A major study developed a dietary index based on these exact principles, and they called it the Empirical Dietary Index for Slower Epigenetic Aging, or EDISEA for short.
The findings were, frankly, profound. When they compared people with the highest scores on this index to those with the lowest, they found an average deceleration of 4.65 years in their epigenetic age. They had quite literally put the brakes on the aging process. So what does a diet like this actually look like? Well, the EDISEA data gives us a very clear picture. You see foods with a positive score, the ones linked to slower aging, include things like fruits, tea, and leafy green vegetables. All of these are excellent sources of diverse fibers and polyphenols to feed that microbial pharmacy. And on the flip side, the foods with a negative score, the ones linked to accelerated aging, include processed meats, refined grains, and with the strongest negative link, beer. And this diagram, using data from the famous Framingham heart study, really validates the entire mechanism. It shows that the known link between a healthier diet and better health outcomes, like lower risk of dementia and death, is significantly mediated by the DunedinPACE score. In other words, a better diet slows the epigenetic clock, and it is the slowing of this clock that directly leads to better health and a longer health span. So, the real key takeaway here is that this whole chain of events, from the food you eat, to the signals your gut microbes produce, to the way your genes are expressed, can now be measured. We've officially moved from looking at broad population data to having the potential for precise personal metrics. The implications of this are, of course, enormous. As the researchers in that eLife paper put it, monitoring pace of aging may inform dementia resilience. This isn't just about trying to live longer, it's about improving our health span, our ability to resist the devastating diseases of aging. And that brings us to the ultimate conclusion from all this data. The rate at which your body is aging is no longer some invisible, unstoppable force. It is a biological signal that we can quantify. And more importantly, it is a signal that we can modify through precise, targeted dietary interventions. So the science has effectively made our aging speedometer visible. For the first time, we can get direct, objective feedback on how our lifestyle choices are impacting our rate of aging. The question is no longer if we can change our pace of aging, but what we will do to control its speed. Now, if you're interested in quantifying your own pace, the DunedinPACE test, which is commercially available as TrueAge, is the tool to use. We put a link for it down in the description.